Biochemistry Immunology Lab in the Qazi Nazrul University, Asansol, West Bengal. His area of expertise, as you can all see, immunology of infectious diseases. He has won so many awards, several awards. It's wonderful, really good, and has a large number of publications in the international journals. So it's a pleasure to invite you. Please make your presentation.
Thank you, respected chairs, and good afternoon to all of you. So thanks to the organizer for giving me the opportunity to present my work. As already Professor Kaur said that lymphatic fibrosis is coming back. Yes, it is true. So my work is to present the possible underlying mechanism of the immunopathogenesis of Bancroft's filariasis. So what is Bancroft's filariasis? Because in India you will find 3 parasites that can infect human to cause filariasis. One is Mukherjee Bancrofti, second one is Bugia malawi, and third is Bugia timori. But amongst the filial cases, 90% are due to the Mukherjee Bancrofti, and this form is known as Bancroft's filariasis. So currently 44 countries across the globe are infected with lymphatic filariasis, and our country, India, it is contributing more than 40% to the global disease burden of lymphatic filariasis. But the current treatment regimen is not sufficient to control this particular disease, this parasitic disease. So immunotherapy could be an option. So if we look at the immunopathogenesis of this particular disease as per the available literature, so when this parasite enters into the human blood through the vector-mediated transmission, the parasite interacts with the number of immune cells. Out of these, the monocytes and macrophages are the prime cells. And when this parasite interacts with these immune cells, so parasite changes their morphology, that macrophage changes their phenotype from M2 to M1. This M1 of the pro-inflammatory macrophages that releases different pro-inflammatory cytokines that can activate different types of immune cells present in different parts of the human body, and that contributes to the overt immunopathology of this disease. So my plan was to purify the particular factor or the particular surface antigen that is present on the surface of the parasite, or precisely on the seat of the parasite that causes the inflammation. For that, we did the night blood sampling, because this parasite only comes in the blood during the night. And so we took the parasite and then we prepared the sheath lysate, that means the surface antigen preparation we have done, and we purified the actual factor through a series of chromatographic procedure. So what we did, so initially we treated the macrophage cell line, that is R264.7, with the sheath lysate, and we found there was upregulation of the TNF alpha, that is a cytokine, and we actually tried to confirm what kind of material it is it a protein or not. So we treated the lysate with the trypsin, the protease, and we also prepared the heat inactivated fraction. And in both these cases, we have found that yes, the material is protein in nature. And then we perform ultrafiltration, and we have found that a fraction of more than 50 kilodalton is effective in inducing the inflammatory response. And finally, we purified the protein through para-aminophenyl phosphorylcholine chromatography, and this protein appears as a 70 kilodalton antigen. So this protein was formally identified through multi-top mass spectroscopy, and it was found as a bestrophin homologue of the free-living nematode, that is C. elegans, available in the worm base. And this protein, it possesses 4 transmembrane helices, and it appears to be a membrane protein. We developed the anti-cell against this particular protein, and we have found that this is located on the surface of the microphage. Now, we wanted to study the mechanism. So, as we have seen that there was upregulation of the 3 important pro-inflammatory cytokines, and these cytokines are the product of NF-kappa B signaling. So, we have found that yes, NF-kappa B activation was there, and in this case, the NF-kappa B activation was routed through the canonical pathway. Now, this NF-kappa B is the transcription factor that gets the signal from the toll-like receptor, and we have found that there was upregulation of the toll-like receptor 4. And when we knock down the cell with TLR4 siRNA, we have found the diminished cytokine production. That means this particular pathway appears to be dependent on the TLR4 and kappa B pathway. So the next question, and important question, was that whether this particular protein acts as a ligand for TLR4. For that, we tagged this protein with FITC, and we did the immunofluorescence. And simultaneously, in another set, we treated the macrophages with anti-TLR4 antibody, and here in these green dots are actually telling that this protein binds to the membrane TLR4, and this was also confirmed with co-immunoprecipitation, and then we studied the protein-protein interaction with surface plasmon resonance, and we have found this protein possesses high degree of affinity with TLR4. And with bioinformatics analysis by the molecular docking, we have found this protein actually binds to the extracellular domain of TLR4. That means this bestrophin antigen present on the surface of the filial passive cellular bank of T interacts with the human TLR4 to induce NF-kappa B, and this could be the reason behind the inflammation. Now, one of the very important aspect of this filial immunopathogens is that the mosquito, filial mosquito, mainly the Q-lex mosquito, it does not release the parasite directly to the bloodstream. It actually deposits on the skin, and then parasite migrates throughout the skin layers where it interacts with the dendritic cell. So we wanted to confirm whether this particular antigen bestrophin is involved in the activation of human dendritic cell or not. So unlike the macrophages, we have found that the protein is actually induces the both maturation and activation of human dendritic cells. And this was also confirmed by the full cytometry data as well as the cytokine analysis. And in this case, that means the dendritic cell activation induced by this bestrophin protein was also TLR4, because when we added the TLR4 pathway inhibitor, that is CLI-095, we have found that there was almost a negligible level of inflammation. And in this case also, we have also found that this protein, that is the bestrophin, it binds to the DCTLR4, and it was confirmed by the flow cytometry and immunofluorescence and the co-immunoprecipitation data. Now, when these dendritic cells, they get the exposure of bestrophin, what kind of T cells T cell response they actually elicit. So we have found here that these MFP educated dendritic cells, they elicit Th1 and T17 response, and also a very minimal degree of T regulatory cells, and it was also confirmed with this. So therefore, the filial surface antigen, which I termed as the novel ligand of TLR4, it actually binds to the TLR4 of dendritic cell, and it causes the release of the cytokine from the dendritic cell, as well as the exposure of this particular protein to dendritic cell causes the expansion of the Th1 and T17 subsets. So this MFP TLR4 interaction, that means this filial protein TLR4 interaction, it could be utilized as a developing intervention strategy. So we actually discussed with our chemistry collaborator from Bose Institute, Professor Anup Misro, and we have found that in the literature and also in the natural product chemistry books, there are so many quinolones and sulfonamides. These are having anti-parasitic activities. So my plan was that if we can combine these two particular scaffolds, we can develop effective antiviral drug that can reduce the parasite load, and it can also reduce the level of inflammation. So we got a particular molecule, there is a quinolone fused cyclic cyphomamide, and this particular molecule can act as both inflammation reducer as well as anti-parasitic drug. But the in vivo response of this particular drug is very poor, so we derivatized this particular molecule with several derivatizing agents, and the 3E, that is a dihydroxy phenyl derivative of this quinolone fused cyclic cyphomamide, it was found that it can induce anti-inflammatory response in the macrophages when we treat the macrophages with this particular compound, and it can bind to TLR4 at the same position where the MFP binds. That means this particular quinolone fused cyclic cyphomamide, it can be a future therapeutic. Although this is a very preliminary study, we need a lot of studies regarding its toxicity and bioavailability. And another just finding, very preliminary finding I want to share with all of you, that is, we developed the anti-sera against this particular protein bestrophin, and when we treated the rat with this particular protein, we have found that this the immunization with this particular antigen, it can reduce the parasitemia in a very efficient way. Not only that, it can reduce the inflammation, as well as if we treat the Uchelia bancopti with this particular serum, and if we inject in the mice, it can resist the mice to develop inflammation. So we can develop vaccine from this. So if any collaborator extend their grateful collaboration, it will be very helpful for us. So with this, So these are some of the publications that we have made, and I acknowledge all of my collaborators and mentors and all the funding agencies for their funding. And thank you. This is my small group at Kathmandu University, and thank you all.
Questions, please.